Hello, everyone. My name is Hunter Lare, and today I will be discussing the article by Chion and Associates that discusses whether philotranscriptomics is as reliable as phylogenomics. Getting into a little bit of background, genomics and transcriptomics can both be utilized to generate phylogenetic trees. So the genomics element of that would be to utilize the whole genome to generate the phylogenetic trees, while phylotranscriptomics uses RNA or the transcriptome sequences instead. There are some pros and cons to utilizing transcriptomics to generate phylogenetic trees versus genomics. The most obvious pros are that utilizing the transcriptome is cheaper, quicker, and more readily available for researchers. There are some cons, though. Those cons are that you will only be able to capture the genes that are expressed. So the RNA that is expressed from the DNA will be what you're capturing there. Gene expression varies between tissues and species. And then identifying orthologs can be challenging, which we'll get a little bit more into the ortholog topic here in a bit. Okay, so getting into methods here. In this study, the researchers really wanted to find out if phylotranscriptomics can produce results that are just as accurate as phylogenomics. And they tested this by examining two groups of organisms. They looked at 22 mammals and then 15 vascular plants. For each group, they built the evolutionary trees in two ways. One was built on the full genome sequence, so genomics there, and then the other was built on the transcriptome sequence. And then they went ahead and compared those trees. There are some more details that we'll get into. So a critical step for both of these methods was the identification of orthologs, which we talked about a little bit before. Orthologs are genes in different species that come from the same gene of a common ancestor. And then comparing the same genes across species can help ensure that you're building out the evolutionary trees accurately to reflect the true relationships between the species. So identifying these orthologs accurately is key here in this study. So there were various methods that they utilized for identifying orthologs. You can see them listed here. For the genomes, they used OrthoMCL and OrthoFinder. For the transcriptomes, they used 5 different methods, and then they compared these methods later on for accuracy with the end result. So I think it's really key that they did that because if you're not accurate in finding the orthologs, it can make it look like the transcriptomics is not as accurate in generating the phylogenetic trees than genomics, but really it was just an error or in inaccuracy in the ortholog identification method utilized. So they used the 5 most common here for the transcriptomes. It was hamster, the YS method, Yang and Smith. And then you can see the other 3 listed there as well. So as they went through this study, they compared the accuracy of those 5 within the end result. And these are the methods that were utilized for tree construction and tree comparison after the genome and transcriptome were analyzed, and the orthologs were identified utilizing the methods described on the previous slide. What did the researchers find? So the main result was that the success of phylotranscriptomics compared to genomics depends very, very strongly on how the orthologs were identified. The YS method, which is very strict and precise, produced phylotranscriptomic trees that were almost identical to the trees that were built with the whole genome data. However, if like the hamster method was utilized in comparison, that is less strict and more errors were introduced. And it led to more differences in the trees when compared to the trees constructed utilizing genomics. Another interesting thing that they found was that the tissues used for the transcriptomes didn't have a major impact on the accuracy of the trees, as long as, again, like I said, the orthologs were properly identified. So that's really good news because it can be difficult to um, find the same tissue in each species or identify the exact same tissue. So knowing that you don't need to do that to get the accurate result is a huge win of the study. So yeah, in conclusion, really overall, the study had a lot of strengths, but it did show that phylotranscriptomics can be reliable and cost-effective alternative to phylogenomics. But that is only true if the process of identifying orthologs is done very carefully and rigorously. Getting into some strengths and limitations. So the study had several strengths. It was empirical, it was based on real data, and it included analysis of both mammals and plants. Um, the researchers also compared multiple methods for detecting orthologs, which, like we discussed in the findings, that ended up being a very, very valuable analysis. Because you can, one, compare the different tools available, but also you can recognize that it is not necessarily genomics or transcriptomics that makes the tree accurate, but it's actually the method of ortholog identification. Um, and so again, they demonstrated that phylotranscriptomics can be a viable alternative to genomics if the proper methods are utilized. Speaking on the limitations a little bit, however, like I said when we talked about the pros and cons, one of the pros to using transcriptomics is that it's cost effective, it's more easily accessible. But if you have to go through this really thorough method for identifying orthologs, that kind of dampers that benefit a little bit because it's not like you're able to do this like really fast throughput and analysis quickly if you have to be very particular about the way that you're identifying the orthologs if you're using transcriptomics vice genomics. So it doesn't necessarily make it as lengthy of a process as it would be with genomics, but it does damper the impact of that benefit a little bit or that pro a little bit. Um, and then another obvious issue or limitation of the study is that transcriptomes only include genes that are expressed or that are currently being expressed at that. So if important parts of the genome are not active at the time of sampling, they might be missed, and that might induce some error as well. And with that, that is the end of my individual presentation. Thank you for listening.